Okay. What is a coat? Coat proteins, as I said, are peripheral membrane proteins. They're soluble in the cytosol. So I said that the cytosol is full of proteins. We talked about actin and tubulin. As we add proteins that are in the cytosol, they're full of coat proteins too. So and they're all mixed together. It's a soup of proteins that are together. So the COP1 coats are proteins are monomers that are floating around here. The COP2 coat proteins are here. The clathrin and other coat proteins, they're all floating around right here. And this shows an EM of a COP1 coated vesicle. You'd have to be an expert to distinguish this vesicle from a clathrin coat or a COP2 coated vesicle or any other kind of post vesicle. I couldn't do that. But I can see that it's a coat vesicle. And this is what I'm looking at. This is in the cytoplasm of a cell. And you can see this sort of circle right here. That's the membrane bilayer of this vesicle. It's the hydrophobic core doesn't have a lot of protein in it, and so this osmium electron stain doesn't pick it up, and you can see that vesicle. And then this is the cytoplasm. You can see a bunch of proteins here, and you can see that a bunch of proteins right here. So that is the coat, and it is formed, this is telling me it's COP1 coat. So COP1 proteins that were around here have fashioned this coat. Inside that vesicle, that's all the proteins that this vesicle is carrying to the next compartment. So the cell is full of coated vesicles like this in the cytosol, bringing COP1 coats going to their, coated vesicles going to their destination, COP2 coated vesicles going to their destination, and the two kinds of clathrin coats going to their destination. So clathrin is not only my favorite, but it's also the oldest one discovered. It was purified by Barbara Pierce in the 70s, a scientist in England, and she purified it and then did a stain right here where she could look at the individual proteins. And what she saw, she called this protein a triskelion because it has this really characteristic shape of sort of three legs intertwined together. And we know now that the clathrin triskelion is made of three heavy chains encoded by a heavy chain gene in combination with three light chains that are thought to regulate the protein. This is the most studied of the coat proteins, although we know a lot more about COP1 and COP2 coat proteins, the ones that were discovered later. And it is found as COP1 and COP2 in all eukaryotic cells, whether we're talking about rat cells or yeast cells or plant cells. All eukaryotic cells have clathrin in them. It's that fundamental for protein traffic. So she purified it, and then you could actually reconstitute the coat assembly process. It'll spontaneously form in the test tube. It's been studied structurally by other people, so this is shown in vitreous ice, and then color-coded to show you how the clathrin coat protein spontaneously forms these hexagons and pentagons. That's what the coat looks like. This spontaneous ability to form this soccer ball kind of structure that makes it able to assemble like that onto membranes and make a vesicle like this. So this is an actual picture from my lab, the only one I'll show you here. This is one we did some years ago where we tagged the clathrin coat protein with GFP, and then we looked at a cell right here. So here's the cell. And we were surprised to find that the vesicles move around, and we knew that the vesicles moved around in the cell, but what GFP told us was that these vesicles can move really pretty long distances in the cell. So you're looking at the vesicles that are forming from a membrane and then whizzing around in the cell, even going from one side of the cell to the other side of the cell. And again, that was a surprise. These vesicles are transient because the coat will form onto a membrane, fashion a vesicle, and then we'll see that the coat falls off of the protein. Okay, so you can tag it with GFP, you can study the structure, and Tommy Kirkhausen, who's at Harvard, actually got together with someone at UCSF to make this movie, and this is an animation showing his discovery. So whereas when it's sprayed onto an electron grid, you saw the triskelions, and they were thought to be flat. he realized by doing other kinds of experiments that it's actually a three-legged puckered structure right here. And he was able to model what's probably going on in cells as clathrin forms onto membranes and fashions a vesicle. So what you're looking at right here is his animation. It's as if you're in the cytoplasm looking at the plasma membrane. So in the cytoplasm, there are a lot of these triskelions that have this puckered. You can see the three different legs right here. And it looks like they have the structure that he did show, also showed they sort of have balls in them. So the triskelions in the cytoplasm all come together. So we have triskelions floating around in the cytoplasm, and they get together with other ones, making this hexagons and pentagons, pulling the membrane in. They assemble onto appropriate membranes, and those triskelions all come together, eventually forming a vesicle right there. Eventually pinching off from the membrane and capturing membrane in there, right there. And the interior will have will have a bilayer, and then it'll have all the proteins inside. So, probably not turquoise in real life. I think it's a really pretty movie. Okay, so how do these proteins form right here? It turns out that clathrin-coated vesicles, I said there were two different kinds, form on the plasma membrane and the trans-Golgi network. So what makes the plasma membrane vesicles different from the trans-Golgi network? Turns out that the clathrin coat is not one kind of protein, there are two kinds of proteins. In addition to the clathrin triskelions, there are other proteins that are called APs, or adapter proteins, or assembly proteins. So APs because that could stand for adapter protein or assembly protein. And the way these work is like this. So clathrin is again found on the, clathrin coats form on the plasma membrane or on the trans-Golgi. Those are for the purposes of this course, we're going to say just those two locations. It looks like the story is more complex. But they form on the plasma membrane or the trans-Golgi network. And this is how they work. So let's say this is the plasma membrane right here. And this is the outside of the cell below in blue right here. And this is the inside of the cell, the cytosol right here. So this could be a receptor. This might be the LDL receptor that we've studied before. And this receptor binds in red LDL particles. Those are cholesterol particles. And the LDL receptor is going to be internalized inside the cell in these clathrin coats that form on the plasma membrane. So this is how it works. In dark green is meant to represent the clathrin triskelions, labeled here. And these sort of little eared structures are the AP proteins. And the tail, the cytoplasmic tail of the LDL receptor will bind to the AP protein right here, and then that will bind the clathrin coat. So cargo receptor in our example right here is the LDL receptor. It's binding to LDL, and then the LDL receptor itself will bind the AP protein. I said one of the names was assembly protein because it will actually promote the assembly of clathrin. And so you get more APs binding LDL, binding more and more clathrin, and together those get together and bend the membrane so that will eventually form a vesicle with some other regulatory proteins we're going to leave out that have this final pinch. But they sort of bend the membrane, bringing this bilayer closer and closer together, so it becomes less energetically favorable to bring these together. And these bud off, so we end up with a clathrin-coated vesicle containing receptors, like the LDL receptor, the cargo that the receptor is binding, and then it almost immediately, the coat proteins fall off. So we end up with a naked transport vesicle that will go to the next compartment. The peripheral clathrin and the APs are peripheral membrane proteins, so they'll recycle back to make, so they're free in the cytoplasm again, they'll recycle back to make another vesicle. But this transport vesicle has trapped now the receptor as well as the cargo that's bound to it. So I've used the LDL receptor as an example right here with the cargo, But there are many receptors that use this clathrin-coated process on the plasma membrane. LDL receptor is one of them. The insulin receptor is also internalized by clathrin-coated vesicles. The EGF, epidermal growth factor receptor, is another one. Ferritin's another one. So there are many receptors that will go in through clathrin coats by binding the AP proteins and then binding clathrin and being transported. And I think that's where we'll end today. So we've ended with the clathrin coats. In the next class, we'll go through we'll talk more about clathrin coats, and then we'll start talking about other coats.